Hey everybody. Um, okay, so let me finish up the slide before this was the one with this one and how you don't need antibiotics with viruses. So on this one, it's talking about viroids. Viroids are the smallest known particles that are able to replicate. A viroid is a short, circular, single strand of RNA that does not have a capsid. And remember, a capsid is part of it. For every virus, you need a genetic material, a capsid, and maybe a protein coat. So this one does not have a capsid. A viroid infects plants such as coconuts, potatoes, and oranges. All right, so the next one is a prion. Prions are made only of proteins. It causes the misfolding of other proteins. So it results in diseases of the brain. If you've ever heard of mad cow disease, this is a prion. And prions are, you get prions from, humans get it from basically contaminated food or eating food with nervous system material in it. So, like eating brains or part of the spinal cord. So sometimes it can come from like different tribes that eat like monkey brains and stuff like that, or eat I can't remember what the term is, but when they just eat parts basically of animals, that can cause it. Also, if a butcher was butchering a cow or a pig or something like that incorrectly and got some of the spinal cord in it or something like that, could also cause prions. So this one, in late December 2003, a single cow in Washington state was diagnosed with bovine spongiform encephalopathy, also known as mad cow disease. The research shows that mad cow disease is caused by prions, which is an infectious protein particles that do not have a genome. Prions also cause a number of other I can't say that word right now. Uh, Degenerative brain diseases, including Scrapie and Creutzfeldt-Jakob disease, or KURU. Okay, so basically, don't eat brains.
Dr. Prusner coined the term prion to denote a protein infectious particle. Now we know that normal prion proteins are abundant in the body and are found in the membranes of many cells, including neurons. As with other proteins, they're produced in the endoplasmic reticulum, processed in the Golgi apparatus, and transported to the cell surface. And as with many other surface-bound proteins, they're engulfed by endocytosis and degraded within lysosomes. The normal cellular prion proteins are designated by the term PRPC. The three-dimensional structure of these proteins is predominantly made up of alpha helical configurations. In prion diseases, that configuration changes to become predominantly beta-pleated sheets. The abnormally folded prion molecule is designated PRPSC because it was first identified in Scrapie-infected sheep. An abnormal prion molecule can induce other normal prion proteins to adopt the pathologic configuration, thus leading to a geometric increase in the number of abnormally folded prions. These molecules aggregate together. They're resistant to proteolytic digestion, and so they can accumulate within lysosomes or on the surfaces of the cells. They're also deposited in the extracellular space and can be stained with an immunostain using an antibody directed against the prion protein. In this section, the brown areas represent deposits of prion protein in a patient's brain.
That protein, when it accumulates, is what causes the disease. The malfunction of the nervous system is really the result of PRPSC accumulation.
Don't worry, I will not ask you any of that, but that was just talking about prions. So, I think that is it for the chapter. So tonight, or later today, that you're going to have to do a study guide. All right, bye.